Good morning, honorable judges. Today, the topic of my report is Antimicrobial Peptide Rex-3 gamma promotes regeneration of intestinal stem cells after injury. Here are the 4 aspects. Nuclear safety is a vital component of national and international security. As is allocated by China, using nuclear energy peacefully is essential for global development. However, the rapid development results in progressive demand in medical prevention and treatment of ionizing radiation damage. IR can cause types of radiation injuries, especially in the intestines. A high dose of acute exposure can lead to intestinal necrosis, which is life-threatening. But currently, radioprotectant is the main way to prevent IR damage. But currently, it cannot meet a requirement of definite target, high efficiency, and low toxicity. It still remains one of the key problems that need to be solved urgently in the field of radiation medicine. In previous work, TAS plays a key role in intestinal regeneration, bringing new thought to the radioprotectant invention. Antimicrobial peptide AMP are a kind of small molecular proteins that are widely found in nature. It has a unique antimicrobial mechanism and can be a potential target for the novel antibiotics to answer the question of AMR, which is antimicrobial resistance. And it is also important to the gut homeostasis, one of which is Rex-3 family. Our research showed that the activation of the Tau signaling pathway could upregulate the Rex-3 gamma's expression. Thus, we tried to identify the key role of Rex-3 gamma in protecting against IR-induced injury. Here are the methods. We used a single-cell sequence to screen differential expression genes to identify Rex-3G. Then we systematically discussed the radioprotective effect and mechanism by using recombinant protein, KL mice, intestinal organoids, and sequencing technology. Here are the results. First, we screened new target for radioprotectant through multiple transcription experiments. LGR5, Rex-3G, and Rex-3B were found to be highly enriched. The single-cell seq found that Rex-3B and Rex-3G were widely and highly expressed in the intestinal epithelial cells, especially in the IICs. In addition, Rex-3 gamma expression was the most upregulated after IR exposure. Thus, Rex-3 gamma might play a protective role in IR-induced intestinal injury. Next, we discussed the intestinal radioprotection of RM Rex-3 gamma. By establishing TBI model, that is, total body irradiation model, we found the relative valence and the relative depth of crypts were more better in the treated group. The IF results show that the treated group have more Ki67 positive and OLFM4 positive cells in the crypts, which means RM Rex-3 gamma could promote IIC's regeneration. Meanwhile, FCM results show that Rex-3 gamma increased the number of LGR5 positive IICs in the crypts, and QRT-PCR results show that it significantly upregulates the expression of key markers of IICs, including ASCL2, which was consistent with the result of intestinal organoids. It is indicated that it can promote IIC's regeneration in vivo and in vitro. RNA sequence was conducted to explore the mechanism of RM Rex-3 gamma. The results showed the DGs and GSEA, of which showed the enrichment of YAX data signaling pathway in the intestinal organoids. The phosphorylation the WB results reminded us the Rex-3 gamma could significantly increase the phosphorylation of STATA3 and increase the expression of key molecules like cyclin D1 and CMYC. Meanwhile, FCM and IF showed that it could increase the expression of p-STATA3 in IICs after IR. These results proved that RM Rex-3 gamma could activate YAT-STATA signaling pathway in IICs. Furthermore, STELLA-3 inhibitor static was used to figure out whether the radioprotection function was dependent on the activation of STELLA-3 signaling pathway. All the observations above show that the static blocks the radioprotection in vivo and in vitro. At last, we established the Reg-3 gamma KO mice. We found that the radiosensitivity of which were increasing significantly. And subsequently, we compared the survival rate with changes, pathological results, and clinical scores, all showing that KO mice were more susceptible to IR exposure. They had fewer LGR5 positive and LLFM4 positive ISCs in the intestinal crypts. The deficiency also attenuated the formation ability of intestinal organoids. In sum, it is suggested that the deficiency inhibited the function of ISCs in vivo and in vitro. In brief, we demonstrated that Reg-3 gamma was required for ISC's regeneration after IR exposure, which could be a potential radioprotectant. And we also highlighted how Reg-3 gamma altered the regeneration of ISCs in an antimicrobial peptide-independent manner, that is, STELLA-3 signaling pathway. The road of scientific research is accompanied with loads of obstacles. However, all the efforts pay off and will achieve fine results. We will continue our journey regardless of pain and loss. That's all for my report. Thank you.
Thank you for your interesting research. Do judges have any comments or communications?
I'm wondering about the in vivo model that you are using, the knockout model of the animal. Is it just the organ-specific knockout or the whole body knockout? Because when you are considering the effect only on the small intestine, but if the result is in the whole body knockout, could it affect other organ injury or disability?
Thank you for your questions. Actually, we knocked out the total body because in this presentation we focus on the intestine, but actually in previous research we also did some experiments about the blood vessels and in the bone marrows and the spleens like that, because the radiation damage is quite severe injuries to the human body and to the organic bodies. So we focus many aspects. We just present the intestinal part. Yes, thank you.
Could you please go back to the DEG page, the first of your results? I just wonder.
Which page, sorry?
The first page of your results. Number one, yeah.
Number one, this one?
Yeah. Why do you focus Reg-3G over LGL5?
Thank you for your question. In differential experiment, we screened that LGR5, Reg-3G, and Reg-3B were highly enriched, and LGR5 is in the ISM's markers, and it can promote the regeneration. So we focus on the Reg-3G and Reg-3B, and in the next PowerPoint, we did the experiment in the picture C, especially. The PCR results show after IR, Reg-3 gamma is upregulated mostly compared to other like Reg-3 beta and alpha and 2. It is the most upregulated, so we focus on this. Thank you.
My question is that why do you ignore LGR5?
LGR5. LGR5 is actually is like LLM4. It's widely known that it is stem cells related genes. So we just try another genes. Thank you.